the Medical AI Podcast.
Hello and welcome to the Medical AI Podcast, a 30-minute thrill ride of big ideas. I'm Dr Félix Balazard and I discuss with scientists, innovators and philosophers. AI has the potential to revolutionize clinical trials in many ways. It could make the development of new treatments faster, safer, cheaper and more personalised. One company in this space is Owkin, a unicorn startup based in Paris. And with me is Félix Balazard, Director of Optimized Development Solutions. Félix, bienvenue.
Thank you for having me.
It's a pleasure. By the way, this is the first time I've interviewed another Félix. I'm kind of hoping that we're not going to get too confused and we probably just need to remind ourselves which one is which, right? So, Félix, we're going to be talking about clinical trials. Could you just first, though, introduce yourself and explain a little bit about what Owkin does?
With pleasure. So I'm Félix Balazard. I'm Director of Optimized Development. I've been working at Owkin for 6 years now. And before that, I was doing a PhD in statistics applied to epidemiology. And before that, I was studying math at university. And so when I joined Owkin, we're a small team of 35 people. Now we're 300, more than 300 person working for the company. And so we've changed a bit what we were aimed to do during all those years. It's always been AI for medical research, but now we've turned into an AI biotech. So we've in-licensed our first asset this year, and we're going to be starting clinical trials next year.
Okay, Now, Owkin has been very successful in terms of attracting venture capital. Why do you think that is?
So it's I'd say it's different stories from the beginning and from the last year. So in the beginning, I think we got venture capital funds from more tech VCs, so venture capitalists more from the like Google Ventures and people like that. And in the last years, we've had really impactful strategic alliances with two large pharma companies, so Sanofi and BMS. And there it's really, we attracted those investments because it was at the same time an investment and an alliance between two companies that wanted to, have some really high-level AI to inform their clinical development, their drug discovery.
I see. All right. So look, we're going to be talking about the application of AI to clinical trials specifically. Could you just, though, for the benefit of any listeners who may not be very familiar with how clinical trials work, could you just take us through how that process works and what the ultimate aim is?
So clinical trials are all the tests in humans that are done to a new drug. In order for it to go from simply something we've tried on cell lines and animal models to something that is fully vetted as a safe and effective drug that can be put on the market and available for prescription to doctors. So trials are cut into phases. So phase 1 clinical trials aim to evaluate the safety of the drug as well as the dosing, and it's in a few patients. Then phase 2 studies are going to further study dosing and safety, but they're also going to be looking at first hint of efficacy, of at least efficacy on a biological level to make sure that the drug is doing what we want it to do. And the phase 3 clinical trial is going to be a very large, very expensive trial, randomized clinical trial, where we're going to really make the demonstration that there is a clinical benefit of the drug. And based on this phase 3 clinical trial, sometimes we say pivotal clinical trial, we're going, the drug sponsors, the trial sponsors, will submit a regulatory dossier to get their drug approved and put on the market.
Sure. Okay. So could you also explain, because it will set us up for what we'll talk about a little bit later, the difference between an active arm and a comparator arm in a clinical trial?
Yes. So in a randomized controlled study, you're going to randomize patients to receive either the standard of care plus potentially a placebo or the experimental treatments. And so the active arm is the experimental arm, where the patients are getting the new drug that the sponsor wants to put on the market and wants to study. And the comparator arm is the arm that receives standard of care plus a placebo.
Okay, that's very nicely explained. So before we talk about what you're actually doing in Owkin, can we just go over what the actual problem is that you're trying to solve?
So clinical trials are very long, very expensive, and very risky. So despite the very large investment of time and money, and also it's time taken from patients as well. Phase 3 clinical trials that are those massive $100 million investments over several years, we're still getting a 50% success rate, which means that we're at this stage where we're launching a phase 3 clinical trial. We're basically tossing a coin between success and failure, and that's very it seems like any way we can improve the odds and get more than 50% chance of success is going to be a massive impact.
And I guess a 50% success rate for a phase 3 clinical trial might not to some people seem like such a big deal, except for the fact that they are fantastically expensive for a company, right? And if an experimental drug fails at phase 3, then it can really knock the company's share price, right?
Yeah, I mean, for a lot of the big pharma obviously have the shoulders to handle a phase 3 blowout, and it happens all the time. But for smaller biotech companies that may have only one asset in-house, if they're sponsoring themselves a phase 3 clinical trial, then they're, basically the life of the company is at risk.
Yeah, absolutely.
A lot of the times the biotechs partner before doing a phase 3 trial with a larger pharma.
All right, so now let's talk about what you are doing in this area. So could you explain how you are applying AI to this fairly fundamental problem with drug development?
Yeah, so we are doing basically two things to improve the odds of the phase 3, to improve the phase 3 success rate. We're doing external control arms and covariate adjustment. So let's start with external control arms. The idea is that so when you've done your single arm phase 2 trial, you're trying to see the efficacy of your drug on a small population, not as big as the phase 3, but you can still start to get a sense of if the drug is working or not. But the issue is that when you're only basing your decisions to start the phase 3 on a single arm study, you might get blindsided by looking at historical comparisons that don't take into account the differences in population between your trial and the previous trial. And so that's what an external control arm brings. It's a control arm that's not internal, but that's external, meaning it's not patients that were recruited in the study, but it's patients that have received the standard of care in another setting. It might be a historical clinical trial, it might be some registries, some real-world data, so that's an external control arm. It's something that's, we're not the only one doing it. Far from it. The different thing we're doing, and which makes it a little bit more AI than the rest, is we're using federated learning to do it. Federated learning is basically the idea is to do machine learning in a decentralized manner. So instead of having all data in a central server, in a unique computer, and training machine learning model or statistical models on this centralized data, we're going to be able to do the same analysis as if we had pulled the data while leaving data in different servers. So in our instance, for an external control arm, the pharma company, the biotech company, might want to keep control over its data to have it in its secure IT infrastructure, and the hospital that provides the data for the external control arm might also want to protect the privacy of its patients, as well as keep control over its data. And what we're going to be able to do with federated learning is, while those data are going to stay in the separate servers, we're going to communicate model information, so model gradients, technical quantities that help compute a statistical model in order to perform the same analysis as if we had pulled the data while leaving the data separate.
So essentially what you're doing then is you're saying to yourselves, okay, there is a control group out there already for people receiving standard treatment, and we can just harvest those data using techniques to adjust the data so that it's comparable to what we're collecting in the control in the experimental arm, right?
Exactly.
And how precisely do you use machine learning to make that happen?
So,
without going into too much detail, just broadly. So
we're doing so in order to compensate for the lack of randomization between the experimental arm and the external control arm, we have to use statistical methods such as propensity score matching or inverse probability of treatment weighting in order to compensate for this lack of randomization. And so this is a well-established statistical analysis, but basically in order to perform this analysis in a federated setting where we have where the data isn't centralized, we have to really go down to the model training steps, decomposing everything and making sure we're communicating the right model information in order to perform the analysis that we want. So, the analysis itself is pretty standard. What's in in a sense, machine learning is that we really have to go into the implementation details to break things down and really do the computations that are necessary to perform this statistical analysis in a federated manner.
Now, I'm guessing that there may be some skeptics out there who are thinking, look, the reason we normally have randomization in clinical trials is for a very specific reason, and that is to reduce or minimize the possibility of bias. And bias can creep in all sorts of ways that we don't necessarily understand. And that's why we'd randomize between all subjects into experimental and control arms. Right now, I guess what you're ending up with here is a comparator arm that has all sorts of potential sources of bias that you may not know about, and you might try and adjust for that statistically, but you're still probably going to end up with a control arm that is not really as comparable as it should be had you used randomization. Is that a kind of standard objection in this kind of area?
Yeah, absolutely. So I think with external control arms, we're not aiming to replace randomized controlled trials in all settings. We're really trying to, make better decisions in the clinical development process. There are also some regulatory uses of those external control arms, but you are as good as the external control arm is. You can measure all confounders. You can have great quality data, and really make the best analysis that you can. In the end, you're always relying on an assumption, which is that there are no unmeasured confounders, and that's an assumption that's unverifiable, and that is dealt with by randomization. Randomization makes sure that there are no confounders. So, external control arms aren't going to replace randomized clinical trials, but they can bring value at different stages of the clinical development process.
Okay, I mean, the basic concept seems fantastic, right? Because a drug company the cost of phase 3 trials are fantastically high in many cases, and if a company could almost halve that cost, then I'm sure it would be rubbing its hands with glee, right? So could you just talk a bit more about the potential benefits of this kind of technique?
Yeah, so there are two main uses. So one is for internal decision making. So based on all the data that the pharma company has before the beginning of the phase 3 clinical trial, making the best decision to go forward with the phase 3 clinical trial or to stop and not discontinue the development of the drug for that indication. And, that's where an external control arm can bring value and really bring additional evidence that it's worth going for the phase 3 clinical trial, taking the leap. Another very important impact is the impact that can be had that you can have at the regulatory or payer stage. So, there's basically, I think, two very important use cases. One is rare diseases. So in the case of rare diseases, you're doing your phase 1, phase 2 studies, and by the time you get to phase 3 clinical trials, you might not even have enough patients left who haven't received the drug to actually perform the trial. So the regulators are, acutely aware of that, and they're very open to considering an external control arm, complementing the single arm phase 2 study to give an approval in rare diseases. If obviously if there is a compelling evidence of treatment effect.
Okay, so why don't we turn to the second of your two applications, and that's about reducing statistical noise. So could you just explain what the problem involved here is?
Yes. So covariate adjustment. So so here we're talking about phase 3 clinical trials, so randomized controlled studies, and so when you're doing the efficacy analysis in a phase 3 clinical trial, you're trying to estimate a treatment effect, and basically the more people in your study, the better you're going to be able to estimate this treatment effect. But there's another way that you can improve the precision of your estimation, and that's by doing a covariate adjustment. So covariate adjustment is simply taking into account in the analysis prognostic information about your patients. And so why would that help? It's really a question of signal and noise. So the signal you care about is the treatment effect. The noise is the prognostic heterogeneity of your patients. Some patients have a very good prognosis and will live 10 years without recurrence. Some other are going to die in a month. And obviously, if you give them the treatment, it might change a bit their prognosis, but it's not going to change it that much. Overall, on the population, you want to see an effect, but people with bad prognosis are going to still have a not such a great prognosis, even if your drug has an effect. And so that's noise in the sense that it's not your treatment effect. It's not just a signal you care about. And by quantifying this noise, we can remove it analytically in the efficacy analysis. So it's really about improving the signal to noise ratio. And you can understand this with a metaphor. So imagine you're on the subway, you're trying to listen to music on your headphones. So the signal you care about is the music in your headphones. But there's all the subway background noise that's stopping you from hearing the music correctly. So you can turn up the volume, and that's going to increase the signal, so you're going to hear the music better. It would be the equivalent of having a larger sample size for your trial. Or you can remove noise, and that's what we can get with those new headsets that have active noise cancellation. They're going to compensate for the noise of the subway. That's going to allow you to listen better to your signal, the music. So yeah, it's really the same thing. You want to improve the signal to noise ratio.
So this is quite related to research I've done myself. Now, normally in a clinical 3 phase trial, normally in a phase 3 clinical trial, one is not trying to predict how an individual is going to do on a drug. One is, on average, looking at a treatment effect, comparing the active arm with a comparator arm, right? So those are quite different types of analysis. So so what you're proposing here is something, how new is it in terms of clinical trial analysis?
So, in terms of analysis, it's really, let's say you have a continuous outcome. The analysis you're going to do is a linear regression of your outcome, with an intercept and a treatment effect. So, you have the treatment allocation variable that's going to be 0, 1, and you're going to look at the difference in means between the two groups. And if you add in that linear regression simply another coefficient with your prognostic variable, that's going to basically remove some of the variation of the estimation of your treatment effect. That's going to improve the statistical power.
But when it comes to actual clinical practice, after the clinical trial, it's not like a doctor who is prescribing a drug or not is going to be performing these kind of statistical analyses on an individual basis, right? So I'm kind of, what is the utility of these kind of analyses for clinical practice?
So you just get a better estimation. So it's exactly the same situation, if you want, as basing your clinical practice on a clinical trial that has 200 patients or a clinical trial that has 300 patients. You're going to get more information from the clinical trial with 300 patients, and you're going to get more information with an adjusted clinical trial than with an unadjusted one.
Okay, so could you give us a specific example, Felix, about how this principle has been applied to date?
Yeah, so covariate adjustment is something that happens in all clinical trials. So, for instance, in some oncology clinical trials, you might stratify your population based on ECOG, which is performance status, how patients, fare, are they able to walk? Are they do they need some help to move, or are they bedridden? And so that's a very simple but very prognostic information. And simply stratifying your analysis based on performance status already is a sort of a covariate adjustment. Now, if we go into so that's a very clinical approach to the question, and oftentimes it's not been done in a very systematic way. It's been done based on clinical expertise, not always thinking about maximizing the prognostic information captured by those covariates, those stratification factors. And so already, understanding that that's the potential impact of those choices can, help make better choices. And also, doing this in a very evidence-based way, in a data-driven way. You want to be looking at the data sets in the same indication as you're going to do your trial, and really estimate which covariate is the most prognostic, how much additional statistical power you're going to be able to get, and beyond even this data-driven approach, then there's the opportunities that are new and come from AI and new genomic modalities. And so, for instance, so at Owkin we've worked a lot with images of histology, and so histology is when a patient has a tumor, we do a biopsy or a resection, and then we're going to be able to cut the tumor tissue and put it under a microscope, scan it, and really look at a lot of morphological patterns on the tumor tissue. And what we realized is that you can predict prognosis with more accuracy in certain indications than previous prognostic information that was available in those indications. So, for instance, in mesothelioma or resected hepatocellular carcinoma, so liver cancer, we were able to train deep learning models that take an image and predict the prognosis of a patient. They output a continuous score that is high for patients with high risk, low for patients with low risk, and we're going to basically using that additional information that has been extracted by deep learning algorithms from histology images can help further improve the statistical power of phase 3 clinical trials. And so to continue on that example, we went to see the EMA, so the European Medicines Agency, to discuss this example, those examples actually, and they listened to all the scientific evidence we provided them, and they decided to grant us a letter of support, basically stating that this is a worthy approach, as well as the next steps that need to happen.
Now, I'm thinking maybe one of the objections they might have had is that you're introducing a black box into the analysis, and therefore you might, in some sense, lose control of what's actually going on. Is that one of the problems in terms of their perception?
So they did ask that question, and we addressed it quite fully. I mean, they were satisfied with our answer.
Yeah.
So basically, we have some interpretability built in those deep learning models. So basically, a histology slide is something that's very, very large. It's like a 2 gigabyte image. It's a very high resolution. And so what's happening is we're cutting the images in tiles, in small tiles. And then we're predicting prognosis at the tile level, and then aggregating the prognosis at the individual level. But all this to say that we can actually show the tiles that have been used to do the prediction. So we can show to an expert pathologist, look, those regions are the things that were used by the model to predict the high risk of recurrence or the high risk of death. And in some cases, the pathologist has actually discovered new patterns that weren't known to pathologists. And so indeed, with deep learning models and all models in general, you have to you can have a trade-off between complexity and interpretability. And but yeah, in our case, we've been able to really keep interpretability as part of the algorithm.
Excellent. All right. Well, look, Felix, I think you've given a very interesting account. Last question, and that's about a completely different area. So could you tell us what your favorite example of medical AI is that's got nothing to do with this area at all?
So I think a lot of people have been very impressed by the AlphaFold moment. So the work by Google DeepMind on predicting the folding of proteins, and it's it was really very, very impressive. It was a problem that has been very difficult for many years, and that's been cracked open by AlphaFold. And so my discussions with, so I'm not an expert on proteins or drug discovery, drug design, and the immediate impact of AlphaFold isn't we're not, it doesn't solve drug design basically, but it's still a very large step in the right direction, and very much looking forward to see the new results from those teams.
Quite right. All right. Well, Felix, thank you so much for joining, and also I'll say bon chance, because you're just about to have a baby, right?
Yes, absolutely.
Have you got any names picked out?
I do.
Felix if it's a boy, I'm guessing. Excellent. All the best, Felix. It's been a really interesting podcast, and I wish you all the best for the next week.
It was a pleasure. Thanks for having me.
Cheers, then. Okay, bye-bye.
Bye.
Thanks for listening. Ping me on LinkedIn if you have any ideas on new subjects or guests.